agriculture, biochemistry, post-harvest, and then food science. The staff strength, we are 43 in number, male being 21, sorry, 22, and then female 21. This one is about us. We can pair ourselves up with only one electron floating in the cloud. We have currently 10 on study leave, pursuing their PhD for their masters, and we recently had one PhD person joining us, just about completing, and then one master student also. And the core, that's research technology grades, we are 23 in number, 7 with PhD. Among that 7, we have one at the professorial grade. The non-core staff, we have 3 helping us. One is a contract person, 3 NAPCO persons, and then 22 national persons. The core mandate of this division is to provide scientific services in biology-related technologies. That's molecular biology, dealing with DNA, RNA, proteins, metabolites, culture, biochemistry, that's general biochemistry, science, service, and food science and technology. Our strengths, we have a very diverse expertise in various disciplines, I must say. The Regional Center of Excellence is hosted by our division. We have very well-resourced laboratories here, I must say, equipment-wise. And then the ISO, we are labs are ISO accredited. Currently, these are projects that are ongoing with the various lead scientists. The key challenges and limitations are that there is limited funds for laboratory resources by way of consumables and chemical supplies, and this is posing some threat in the sense that there is a possibility of us losing our ISO accreditation. We cannot reach our full research potential. There is also this lead obviously to limited visibility, because the more we publish in high impact journals, the more visible we will position our institute and the council in general. And then this will also lead to diluted credentials at Regional Center of Excellence. And for some of our young guys that are coming to join us, they have the expertise and they cannot get the resources to really reach their full potential. One of the risks is that they may leave us. And how do we circumvent these risks? The only solution is a reliable, sustained funding. Our division, the DSP Biotech Science and Post-Harvest Division, has huge potentials to internally generate funds for research activities, so we can be able to sustain our activities. Are there any opportunities? Yes. Incessant requests from organizations, researchers, students, etc. wanted to come and to contract us to conduct biochemical analysis. This is a case in point. Just recently, we had this request. An exporter of soybean who wants to conduct some analysis on these various specifications, and looking at all these, we can run them. Among one of the specifications, the detection as to whether the soybean that he wants to export is a GM or not. So what we must do as a division is to strategically position ourselves to offer specialized contract laboratory services in biochemical analysis, tissue culturing for ceiling generation, for example, GMO testing and certification, because with the bill passed or about to pass, definitely we will be having these GM. There should be the possibility for us to differentiate between the non-GM and then the GM crops. And then also seed testing and certification. We can lead in policy, so this will be sort of mandatory. Our lab can position ourselves in such a way that seeds, those that are selling seeds, whether breeder, foundation or certified, would have to get their products tested and then certified by us before the permission will be given. And what are the key requirements? Here, startup funds to procure lab consumables and chemical supplies. And once we've had these supplies, we will then have to develop and optimize as well as validate our standardized protocols. Then we can go ahead to develop a dedicated website where we will post and advertise our services to the world. And these are some positions. Donations from funded projects are startup, and the second one will be to develop a tailor proposal to secure seed funding to generate resources in a sustainable manner. So for the second proposition, this is what we are going to it's in preparation, and we intend submitting, plugging it at the Secretariat for funding. That is sustainable resourcing of the biochemistry, molecular biology labs to engender sustained generation of funds to provide research and clientele based services. Now for research activities, I am dedicating myself to lipogenesis enzyme. Here I'll say the I helper as well as a billion. Why so? These enzymes are involved in numerous metabolic pathways in plants, leading to the biosynthesis of flavor compounds in fruits, vegetables, as well as odor. The flavors may not be pleasant. Signal molecules for physiological activities, plant regulators, antimicrobial compounds, etc. But what they do is that they use these three main polyunsaturated fatty acids, that's linoleic acid, linolenic, and then arachidonic. And these fatty acids either may be free or they may be bound to membranes. If they are bound to membranes, then it means, I mean, this will lead to destruction of the cell, and this will cause cell death. The reason being that the early plants that are formed, they are highly oxidizing in nature, so we refer to them as reactive oxygen species. And in oil seeds, normally what they will lead to is this rancidity and loss in seed viability. This is one of the main reasons why we have most of our oil seeds having germination problems over storage. And how the plant is able to adapt to these is that they use antioxidants. And if these antioxidants are used, because I mean they are normally macronutrients, it will mean that whatever plant that this activity is going on will lose some nutrients. So the objective is 3, to assess the effect of lipogenesis activity on groundnut seed germinability and groundnut oil quality attributes over storage. And second, to determine the influence of fertilizer type on lipogenesis activity and groundnut seed germinability, and then also to determine the effect of lipogenesis activity on physiological and physicochemical attributes of rice seeds stored under different packaging and environmental conditions. The work last year was actually devoted to making sure that we develop the various protocols that we will need because of this issue, this COVID issue. The one chemical that we needed most for quantification, which is very, very critical indeed, delayed. So we have not been able to finalize this activity, so we'll go on. So for this year, activity number 1, there will be 4 main activities. Number 1 will be assessment of lipogenesis activity on groundnut oil quality attributes and effect on seed germination and storage. And here the specific objectives will be to establish the relative lipogenesis activity of groundnut varieties, so that we will be able to advise breeds to lines they can select as parents when they are undertaking the activities. To evaluate the effect of lipogenesis activity on groundnut oil quality attributes, that if we are going to store groundnuts for some period, how will the duration affect the quality attributes, and also to determine the effect of lipogenesis activity on groundnut seed viability. The research team will be biochemistry and post-harvest group, and then the oil seeds group. These are the varieties that we are going to work on. I'm told that there are about 16 varieties at the moment which we can have access to work on. Activity number 2 is investigating the thank you on lipogenesis activity on groundnut seed germinability. Here, this is a topic that our own Sylvia Archer will be working on for her masters. The notion being that metal ions serve as cofactors for this enzyme, so they promote the activity of this enzyme. So here what we are going to do is that three varieties that have low amount of lake will be used and compared with 3 others that have high lake content. Activity number 3 will be the effect of lipogenesis activity on physiological and physicochemical For the physiological part, that is what Ms. Akule talked about. So that is what she is engaged in, and we will be engaged in the physiochemical attributes. Here the special objectives will be to assess the effect of lipolysis activity on rice seed viability over storage, and to determine the redox activity, that's both enzymatic and non-enzymatic. The team will be the rice research group and then the biochemistry post-harvest group. The fourth activity with method development for the determination of aroma intensity in rice. Here aroma we know is a key preferred attribute in rice. But unfortunately what is happening is that during the breeding process, there are two main methods that are used. One sophisticated, that is GCSMS, gas chromatography, mass spectrometer in tandem, or what they do is that they will subject some of these tissues to potassium hydroxide to sell or chew the grains, and you know this is very, very subjective. And the hypothesis that we are going to use is that the key compound that is responsible for aroma is 2-acetyl-1 pyrrolate, and because of this functional group, it should be able to undergo the iodoform test. So this is what we are going to manipulate to see how whether we will be able to succeed in developing a test. So here the objective is to develop a simpler and objective spectrophotometric based method to assess aroma intensity in rice. For my non-research activities, here they are: teaching, genomics, thesis provision, proposal development, review of scientific manuscript, so on and so forth. And our publications, the first two through a supervision of master's students from Cape Coast University. The third one, which is in the manuscript form, through our own manual that I'm co-supervising. And then the fourth one is data that I generated during my PhD that I've developed the manuscripts reviews. Acknowledgement to Dr. Professor Maxwell, Asante, and the entire biochemistry team. Thank you very much.
Thank you. Next presenter is Dr. Let's give him a round of applause. We have 10 minutes.
Good morning, everyone. I'm actually an Alaska chemist and Alaska environmental chemist with the biotech CEO and post-harvest division. Presenting on the topic evaluation of heavy metals and global mode of machine-made food from 6 selected communities in Kumasi Travel East. And these are my team members. In order of presentation, I have the introduction, statement of problem, and objectives technology that I have. Now, by way of introduction, fufu is a traditional food dish popularly eaten in most parts of Ghana and in some African countries. Fufu is mostly made from cassava and unripe, mature plantain processed into a thick paste with smooth consistency. Processing involves pounding boiled cassava and plantain into thick paste with mortar and pestle. However, this process involves a lot of drudgery and sometimes unhygienic. During this process, the actors involved could sweat into the food, talk, laugh, cough, etc. that gets contaminated. The hands are considered the most important vehicle for transfer of microorganisms from faces, nose, skin, other parts of the body into food. That's according to WHO. Now, the machine we see here is the new and innovative mechanized methods of food that will be introduced to the market to overcome this trend of pounding with the mortar and pestle. Now, the one you see here is the one with the metal. The one that is made of metal. And this one is made of wood, but the metal that does the grinding is inside the machine. The machines are becoming quite popular as of most homes. As a result, most homes in East are now patronizing this new mechanized method. This mechanized method for making fufu has contributed several merits to it. Could also be a means of introducing microbial and metal contaminants into the food if not properly handled. Concerns have been raised daily over the hygienic practices of the food machine operators. Basic good hygienic practices is not adhered to. Improper routine cleaning of the grinding parts and maintenance of the machine serve as a source of microbial contamination and may pose a potential health risk to food consumers, such as myself anyway. Most of the parts of the food milling machine are made of galvanized metal, aluminum and iron rather than stainless steel. This can be a source of toxic metal contamination resulting from metal to metal friction, and also sometimes they also use this metallic sponge, which we normally call in our dialect the sapper. Therefore, it assess the levels of these contaminants and also create awareness in order to streamline the operation of the in metropolis to ensure the safety of food consumption by the general public. Now, the main objective is to determine the extent of human exposure to heavy metal consumption and the microbial of milled fufu. Now, the specific objectives are to determine the presence and levels of some of these metals in locally milled fufu from North Metropolis. To determine the presence and levels of foodborne pathogens in milled fufu, and operate to draw conclusion and provide suggestions for actions to reduce contamination during the process, and also to develop a processing clean manner. Now, the methodology, ongoing collaborative research with the chemistry department. I will provide a baseline for heavy metal concentration in the various fields. If you realize some time back last year, we were all over the place. We were taking samples at the various fields, and then we are working on it, and we'll be coming to our findings very soon. And I will provide a baseline for all the heavy metal concentration in all the fields, because over the years, the practices that have gone on, we assume that probably there might be a whole lot of deposits of this metals on our fields. Now, cassava, that's the double lamicity and ampon, and plantain samples from cassava will be used. Sampling will be done in 3 open markets and 3 residential areas. Now, the 3 open markets have the Ija market, and Kwaso, and also residential areas are the Fiabra, Ofu, and Dynamic communities. Now, the methodology will do the chemical analysis, analysis will be done, meta-analysis will be done using the AAS, and then the microbial analysis will be done, the fecal coliform, total coliform, and then the E. coli will be done at the microbiology laboratories at KNUS. The data analysis will be used in the Genesis 2012. Also, funding has become a challenge, so I've put in a proposal to send to the CSIR for grants, and hopefully I'll get that grant. And these are publications for the year. Thank you for your attention.
Thank you, Mr. Chairman. I'm Michael Akwamaboatum, a food and post-harvest scientist. And this is the overview of my presentation. Introduction, objectives, methodology, publications, and acknowledgement. I'm presenting on studies on dormancy and sprouting mechanism based on physiological activities and proteomics as means of improving yam storage. Post-harvest losses in yam are increasing, posing very serious food security threats in Ghana and West Africa as a whole. Storage loss in traditional storage system can reach up to 50% in the first 3 months and over 50% in 6 months. According to a study in Ghana by sprouting alone could cause about 23% of the losses in yam storage. That's after dormancy break. So this makes dormancy very important, because as long as the tuber remains dormant, it means you can store it. But once dormancy breaks, then storage becomes very difficult or virtually impossible. Studies conducted on yam post-harvest losses have mostly centered on reducing external pathogenic loads and controlling physiological agents that cause deterioration and spoilage, and all this aiming at minimizing losses in yam storage. However, losses due to sprouting is still high in stored yam. A better storage regime and condition, if displayed, will help shelf life and ensure all year-round availability, as well as maintain their edible and marketable qualities. Therefore, the main objective is to generate deeper insight into the mechanism that regulate dormancy and sprouting of yam using proteomic data derived from yam varieties of varying sprouting susceptibility. The specific objectives are to determine the biochemical changes in the yam tuber and how they impact on the quality attributes of yam. The second one is to determine comparative changes in gibberellic acid levels in yam tuber and yam tubers as natural progression. And the last one is to generate protein data to identify key proteins that are associated with dormancy and sprouting. Material generation and storage. 2D data cultivars, that's Puna Labrico, and then 1D alata by Ahwadinfu was planted and harvested at maturity. These yams were studied in a yam bank here in Crop Research Institute. Sample preparation. Sampling of yam tubers was done at harvest and then regularly every 30 days until the 90th day of storage. 5 yam tubers were randomly picked for the respective cultivars at its specific storage points. The tubers were washed under running tap water, air dried at room temperature, and cut into 3 regions. We have the head, the middle portion, and then the tail. The tail was peeled off and about 1 centimeter deep to the flesh of the tuber was cut out to form the outer portion of the tuber, and then the remaining one formed the inner portion of the tuber. A representative sample was obtained by pulling respective regions of the 5 tubers. The respective outer portion and the inner portion of these 2 regions were freeze-dried, pulverized, and stored for analysis. Specific objective 3. I've jumped to specific objective 3 because I'm done with specific objective 1 and 2, and what I'm working on is specific objective 3, and the activities involved in specific objective 3: extraction and cleanup of protein from yam tubers, quantification of the extracted proteins, digestion of the extracted protein into tryptic peptides, and then sequencing and identification of protein by LS-MS. The first 3 activities have also been done last year. So what is left now is sequencing and identification of protein by LS-MS tandem. And this cannot be done here, so it's being done outside. But because again of the pandemic, it will delay for some time, so I'm still waiting on it. So whilst waiting, I'm working on determining enzymatic activity of the yam tuber to support the data I have generated already. And then I'm also involved in the work Dr. Amakwando presented, that is assessment of liposogenous activity on groundnut oil quality attributes and effect on seed germination over storage, and method development for the determination of aroma intensity in rice. These are publications at manuscript level. It's being reviewed, it's under review. Acknowledgement. Thank you.
Thank you. You guys are ready. Our next presenter is Mr. Isaac. Let's give him a round of applause. If you look at the program, Victor is being pushed to the next group, who is on film, and this will be done. And I've just been filming it. Actually, we will present the film actor. So let's invite Faustine. Thank you. You have 3 minutes.
Thank you very much. Please, I'm 49. Actually, I'm not 14. Correct. Thank you very much. This is going to be the outline of my presentation. I will touch on. Sorry. I will touch on the work done. the wrong.
Everything is ready. Can you come raise the sound system? Waiting. 15. The chair is not going to move. The chair is then all the time to go back in. Thank you.
Good morning. My name is Osiki. I'm presenting on the work done in 2020, then the plan yet 2021. The first activity was on development of rice multiplication and dissemination system in Ghana, which was funded by FASI for 3 years. The project team. As background to the topic, rice is a very important food crop in Ghana. It generates a lot of income for many people. One major constraint that we have in the system had been low yields that farmers have been producing in their fields, which is far below the achievable yield of 6 metric tons per hectare. And these low yields could be attributed to a number of factors, and one of it is quality seeds that farmers use in their farms. Then there's also poor access to preferred and adapted varieties. And there's also low technical know-how in rice seed production and inefficient multiplication and dissemination system. So it was in line of this that came into being, and the main objectives were to develop a model for a sustainable rice seed multiplication and dissemination system, then to strengthen the existing rice seed multiplication and dissemination system, and to build the capacity of technicians, specialists, and farmers. And mostly what happens is that when there is a developmental project, we not only build the capacities of those who are in the formal system, and we neglect those who are in the formal system. This project seeks to build the capacity of those who are formal and the informal system. So the project started with an reception meeting, then we zoomed into production of breeder seed at Kwadaso, then we also established a yield trial, then there was a training of rice seed technicians, and we also organized and sensitized innovation platform members in the project community. This was the inception meeting, which was attended by the relevant stakeholders and the project team director was present. And on the breeder seed production in the yield trial field, we started with nursery establishment. Then it was the yield trial that we established at Kwadaso. So the field was properly covered, and the director picked us a vitis, and which he was very happy and he commended it for the good work done. So with the rice seeds, the breeder seed production, we were able to produce 2 metric tons. Now, aside the breeder seed production, we also tried to build the capacity of seed technicians from crop and MUFA, and our deputy director graced the occasion on that day, and Dr. Maso was delivering his lecture on that day. We managed to publish the training and issue that certificate to the participants and the rest persons as well. So the sensitization of the innovation platform members, we try to let them understand the project goals and objectives. So we participated in the communities by letting them take and know they can play to ensure the project is successful. The second activity which we did in conjunction with the Breeders' Production was establishment of a youth trial, and there were two main activities that we tried to impose, but I'm trying to present only one of the aspects, and that is the effect of threshing methods on quality of all rice varieties in Ghana. You know, threshing has effects on quality, and there are various methods that are used by the farmers. Sometimes some of them can thresh by using the barrel, can thresh by using the stick, beat the sack with a stick. And so we try to identify the appropriate threshing methods with minimal effect on seed quality, especially on germination, then to determine combined effect of variety and threshing on seed quality. So we established the experiment using 4x3 factorial arrangement in CRD 3 applications, using variety 4x4 main varieties as Agra, Quetia, Napa, and Sadati, and they have been the threshing methods, which are the hand threshing, barrel threshing, and beating with. So this was the interaction effect on the seed quality, and you can see from the table here, if you look at the moisture content, there wasn't any significant difference, but we can see that the moisture content was very low, which is very good for moisture storage, and the purity percentage here too was also very high, meaning we use a pure seed to conduct seed quality tests, but the effects on the weight. So let us the same figures with the same letters as the means that there was significant difference between them. There wasn't interaction effect on the germination, but with the assertions or the main assertions, there were significant differences. If you look at this point, we could see that the NAPA was significantly different from the other three varieties. When you consider the effect of threshing on germination, we realize that threshing would have the highest germination percentage, and back threshing recorded the lowest. Meaning if you thresh using the hand, you stand to gain of the seed quality attributes. Then the second activity to talk about is the supervision of cold room and processing center activities, which actually involve supervising all the activities ranging from threshing, drying, cleaning, and sorting. And fumigation too was also ensured throughout the period. We make sure that at least twice within a year we fumigate the cold room. Then we also ensured the maintenance of the air conditioners in the cold room, and machines and equipment at the processing center. And we also make sure that we document all the activities that the processing center and supervise quality testing at the work. I was involved in capacity building of technicians, farmers, and my own technicians I work with. So plan activities for 2021. I'll continue with the capacity project by establishing a fund for breeder seed production, foundation seed production. Then we also establish the demonstration plots and the project communities. Then we will look at building capacity of seed specialists. Then organize a community information platform for effective dissemination. Then we organize farmers field days and seminars, develop a memoir on foundation seed, and publish posters and fact sheets. So will be one activity I will be doing for this year to determine the effects of descent and storage period on seed quality of 3 soybean varieties in Ghana. So this will be a research team. So the main objective will be to determine descent effects on seed ability, germinability, vigor, seed health, seed weight, then determine the best desiccant to preserve the viability of soybean storage under ambient conditions and to investigate the combined effects of this and storage on seed quality. So you establish experiments to organize the by using RCBD with applications. Then we will use 3x3x3 factorial arrangement in CRD with 3 applications for the laboratory experiment. And these are the factors that we will consider. So these are the growth and the reproductive data as well as sequence data that will be taken, which data will be analyzed. So this one will be my core activity that I will be doing. So this publication references. Thank you very much.
Last agenda for this session is okay. We have 15 o'clock. You have 10 minutes.
Thank you very much. Sorry for what happened from the beginning. So as I already said, I'm Faustina Ouch with the Post-Harvest Division. I'll touch on the work done in 2020 and then the activities plan and then the few publications. During the 2020, I was involved with the PABRA project, Kafasi Post-Harvest, and my other activities was the SDF Post-Harvest. And under the PABRA project, as a way of promoting the release variety, I'm developing consumer acceptable canned beans from 2 of the release beans variety, that is Adoye and Enepa, and the team is shown there. By way of introduction, beans is known for its richness of protein, fiber and other minerals, but this particular beans has zinc and iron that makes it so special. And as we know, most of the canned beans that we consume in Ghana here is imported, and it's formed part of our salad, and we also eat it with jollof and sometimes with sausage, and then as part of our breakfast. That is if you want to enjoy English breakfast. And one of the products which can help our release beans variety to really go all out there is to be able to develop a consumer acceptable canned beans which has similar or even better sensory attributes than what is imported. If you're able to do that, it will help our beans to go all out there and do a lot of magic, reduce the importation, as well as creating employment and also putting in the pocket of our farmers. So my main objective for this work is to develop a consumer acceptable canned beans from Adoye and Enepa, which is part of the release beans. The beans that was released, there are 4. We have Enepa, Adoye, CSIR and Nshoma. Specifically, to develop a consumer acceptable recipe for release beans and then assess the consumer's preference for the beans when it's canned compared with what is on the market. And I will also study the shelf life of the canned bean products. As part of the methodology, it's still ongoing. So this has been done, though this one has been improved to better status. This is what I use for the century, for the development of the beans. First, I had my beans, I soak it in warm water for 16 hours, and the water was changed intermittently, just to help in the reduction of the cooking time, as well as reducing the fighting that is present with the beans, and also to help do away with the nutrients which can put the threat if you don't process it well. So the beans was soaked, and afterward the water was poured away and it was cooked till it was soft, and then the sauce was prepared from brown sugar, tomato puree, kitchen mustard, and salt. This methodology is what I use, but it has improved to a higher level. And afterwards, I developed 3 different samples. One I did not add any extra tomatoes, but the other ones I added barbecue sauce and extra tomato puree, just to make the difference and see how best consumers or assessors will score them for it to check and see if it meets the standard. So I use these 3 methods, and I conducted a tabletop sensory analysis, which composed of about 8 consumers from different backgrounds, and the attribute that I used was the appearance, aroma, texture, taste, bitterness, and the overall quality of the product. And then it was scored for 5-point scale was used. It was scored whether it was bad, fair, average, good, or very good. And at the end of the day, as I scored the samples which had each tomato sauce to be almost equal to, they had the best scores, the ones with the increased tomato sauce. And the Adoye standard also came up to follow up. And the NEPA standard had a score. And then the ones with the barbecue sauce scored least. And then the NEPA, the texture of the NEPA standard extra tomato was a bit scored least. And it might be due to the varietal difference. At the end of the day, we came out successful with we had an overview of how the sauce, the recipe should go. So it gave us a way to go. And then we saw that the ones with the tomato sauce were scoring good compared with that of the barbecue sauce. And then the Adoye was also came out score better than the NEPA variety. But from the analyst comments, we realized that the outer skin coat of the beans was a bit chewy and hard. So we are still working on it, and we hope we will come up with the best. Other activities that we did during the 2020 was that we met the school feeding managers on how they will include the beans in the menu of the school-going children. In that, we believe that if it's involved as part of the menu of the students, this picture shows the meeting that we had the managers in their office. And this is the regional coordinator. So after meeting them, we brought them, the caterers down here, and we trained them on how to soak the beans so that the cooking time will be reduced, as well as how they can add it, they can use it in place of cowpea. Most of the cow beans that they use, cowpea that they use for the food feeding program. Most of them are imported from the neighboring countries, and we believe that if this bees is able to do what the cowpea is doing, it will help to reduce that importation, and then the children will get the zinc and the iron that they need to boost their immune system. We all know that this COVID, what is very important to have is zinc. So if we promote this among the school feed children, it will help to boost their immunity, and then their mind too can work very well. So we met them after the training. They really enjoyed it. Some of the dishes that we prepared was white, and we saw that the variety, even if you use it without white leaves, the white came out so well. So they were so happy about it. And we know that if this is promoted this way, it pushes the variety so well. So they really enjoyed, the coordinator and everybody. And then we also called on about 18 persons from different institutions to train them on novel products of the bees. We trained them on meat substitute and the milk. And some of the people who were involved was the RIAD. We believe that when they get the concept, they too can train others. And then we had the catering lecturers from the schools, and we had the NBSA people, and we had the church men leaders who can also train the church people on it, so that if anybody wants to pick it up as a business, the person can move with it. So we trained them, and they had a hands-on training, so they really enjoyed it. They've been calling always for the beans, but just that the beans face challenge. This is what I'll be doing this year. I'll continue with the baked beans to the level to get it canned, and then compare with what is on the market. And then I'll also be involved with the rice variety release. Under the SDF project, though the project is ended, we are taking it to the highest level, whereby the training materials which we have developed, we are going to have an online training for the public, and it will boost the scientists to be involved, visibility, as well as giving money to the institute. These are some of the training materials that were developed from the SDF post-harvest. Thank you.
We urge all the other presenters to join for stage for the interactions. Thank you. As they take their seats, the next group is now related. Can you please rise with the coordinator to obtain your presentations? Thank you. So we give them the time. It's time for interaction. And I will give first word to the people of the house. nature, and he was lifting appointed. And he also checked my face. First thing, I know you're being there, but when you're in primary, do some kind like a value addition of our materials. We got that apprehension from Food Research Institute. And then if you are collaborating with them, you know the tell them that we are not taking over the language, but we are implementing. And I also want to know, we don't know what Professor Webb, if you understand the what's going on, and also yes. Today, during this session, I'm going to control the time. So, discussion is for 25 minutes. And by that, as such. Thank you very much. The mandate for C certification is with PQRSD. So we could only collaborate with them. They were given the national mandate. And then, we didn't tell us how we're going to take the full samples. And then we were going to the residential area and then how we're going to take the samples. Mr. Kwame, how do you quantify the extracted protein? And then, so initially we called the processing center. I was aware that the private people were using what have we injected into the C center that will make research from there. Still, we said we had improved the power and methodology. What did you add? I was thinking that it is the sweat and all those and that the fufu. This is the best. This is why the new fufu is unique.
Thank you very much, Mr. Chair. As for my issue, I don't have any questions. Dr. Mari, so which is the better, the pounding with the person or the machine? Because aside the fufu, we don't nail most of our fruits. Even the tomato, pepper, onion, ginger, everything is going into the meal. All these medications that people are selling, the other time somebody said something, the person had opened the capsule which came from Ghana, a herbal capsule, and poured water on it, and there was this very thick substance. And I suspected this maybe engine oil or something. So what are the risks when we're staying as a nation? And what can we do? Mr. SA22, your phone service is small, you're most of the time talking to the screen. You have blank spaces on your slides, especially your background slide. And wondering your works that you're doing. How are you liaising with Mr. Dan as to who is also looking at working in rice seed, if you remember rightly from his presentation. And I also identify with the previous question, how are you because if you've been through these meetings, and I would ask you to sit through till the end of today and tomorrow, and unfortunately the leaders have presented a lot of issues with us. Firstly, the presentation was okay, but we were not still. We were showing most of the time. I think it was even taking energy from us. So please, stay still when we are presenting. We should begins and we pay off the later. I'm concerned how much nutrient do we need in that activity. And your testers, the assessors, we have trained assessors that you use to have the core trained assessors to help you with your various activities in the free product development. Thank you.
Then Christina, actually, maybe I'm speaking from a young man point of view. I was expecting that right from the word we assume that we obey this experiment. We have expectation to do the highest place which is standard like as a check. As we try to develop our common products. So why they want to use that as a check? So that we compare the taste. Or the scanning change the taste. Because it's as if you say you can it and then you compare the goods in the market. I'm not so sure, I'm speaking for a real market, but I expected that we use the highest good goods as a check. But then you know that this is tasting even better than the highest good goods that people prefer. Then we can make a case like this. So that is my question for you.
That's the reason why she is here. Professor Marian
Thank you very much, Mr. Chair. Dr. McCarthy, you said you have the potential to generate income, but you have problem with funding. What efforts are you making to resolve this issue? You can actually. And I think you could have also a few pictures because in your presentation there wasn't TV, snowgrass, there are a few pictures to add some support to the presentation. And the same goes to Mr. Kumabwati. We are trying to describe the YAM, how methodology. Maybe a picture for us to see what actually happens. Thanks, Mr. Chair. I'm going to start people has been benefiting too much. Anyone here? Okay.
In fact, I agree with a minute what Professor Lamte said on the control. We should use something that. And my question is, we said one of the variety had ensuring codes. And you don't need to say that you will be working on it. Working on it in terms of, is it the period duration or you recommending it to the breeder to work on it?
You mentioned the Department of Both and Legon, the preventive. I realized that yes, you were devoting PhD aliphatic is what is so fascinating about your plan? You know your plan. What is it? Growing up, I like this.
Outside. What are we doing in that regard? What is doing because lab person that is national systems accredited. We can lab accreditation. Then we can go over some of these.
Maybe you do some life before you finish the
that the speed unit on can have
I majored in botany
Thank you, Mr. Chair.
zoology. So my
I'm interested in Mr. Kunle's presentation.
zoology
We used to cultivated the yards that you used in your work. I was wondering, did you apply any fertilizers to the yards?
and
Leaf race or will that have implications on your results?
so typically
The latest topic when it comes to the application of fertilizer drain in nitrogen, what is your take on that?
and then
You also mentioned this as a potential substitute for wheat. I'm very interested in that. Can you
Out
throw some more light on it for me?
that has something to say. Like you first mentioned.
Thank you. One question goes to Fosti on the summer beans for water.
So what was actually
I'm
on
asking whether the canteen was involved in the demonstration, the Bush canteen and other canteens, because we go out and people ask us a lot of questions and we are not able to tell. If we are not,
my mother a nurse.
how are we planning to improve them? Because I know they do good work. So if you have good things working, it should start from
I
home.
handled myself.
Thank you, Mr. Chairman. My question also goes to Fosti, and I think I have a concern with Priscilla too. Is it you who is going to improve the seed coat, or is there this? The next one is that you mentioned about the imports of cowpea. In fact, in my work, I realized that this watch seller says that it's seasonal, so it's not all the time that they import those cowpea. It is when the local ones are not in season. So if the baked beans is going to replace it, then fine. But then whatever it is, they will still import those ones. And then they also say that those imported ones are more, the cooking quality is faster than what we have here. And so I think that the breeders should look at it and improve on the cooking qualities, and then we promote it, our local ones more, so that it will always be available for the watches sellers and then consumers to use. Thank you.
Thank you. One question.
Question from Nigeria. Please, I want to know. A lot of you are saying that you have production rights. Where do you keep your production rights? It's a problem. This is quite research library. And people come from you, they don't get what you say they will produce. So I would like to know where you keep them, so that you can come for them.
Thank you. I'm taking the last question from Jonas. And you must be snappy.
Well, thank you very much. My name goes to Dr. Ochi. Some time back, we were working on affluence management. Because I didn't know anything about that. Can we know how far would that work? And the other one goes to the head of the and that is the first one. We got some equipment for the seed unit. Washing it and others with the hope of generating some for the institute. Are you aware of those equipments, and if so, how can we collaborate to commercialize them? Thank you.
Now the chairman will come out with his comments before you take your accreditation. Better. You say I've done some work on how to generate revenue. Yes. If you look at Faustine's presentation, I was reserving this comment for commercialization. But my take is that we must, as an institute, target the private sector schools, business schools, with some of our products. And when we are able to take them as our clientele and we supply them regularly, the new go out for the government to now come and enroll us onto the school feeding program. That is my take on that. Dr. Otri, your microbial work, like I said, that's what gives the fufu the taste, the unique taste for Ghana. But when you are doing your methodology, you started mixing up that work with collection of samples from our research fields. So I got confused. Is it the fufu at the research level or at the machine processing level? Policy on strategic policy. Director, I still come to you. I've been championing this issue for a long time now in the institute. The biochemistry lab has one of the best equipment in this country, especially within this building. When the food research guy came, when we were doing this ISO accreditation for the lab, they saw the equipment and said, look, we have more than they have, yet we've not been able to accredit any test in that lab. And in that lab, the protocols are already standardized. They are in the books. So it's just a matter of developing the manual. Because every year, those who go for certification from Standard Authority and Food and Drugs Board, they come here to do they want to do their test before they submit. And this is what we can do for money. So I think we must look at that strongly as an institute. And Faustina, you are doing this thing just opposite my door, but you always produce some pediatric doses of the wax, which when I get on my tongue, I can't swallow. Me, I want it in bowls, so please involve me in the test. Thank you. Can you now respond? Please, not.
Thank you, Chair. Director, you asked about, you talked about the fact that, there is an existing business plan. I'm not privy to this, so I'm going to do a follow-up and see how we can merge with some of the ideas.
Hello, this is CRI TV Beyond the Research, and this is the maiden edition of Women of Science. I have here a noble woman who is going to be our guest for this maiden edition. Here in Ghana, mention GMO and
mandate of certification and testing with the PPRSD. That is well noted. We'll do well to collaborate with them. But the issue is that we think it is not all the tests that they have the expertise to conduct. So we are going to collaborate with them and then see how we can bring them on board and move ahead. Prof. Betch, efforts being made, to raise funds. That is precisely what we have in mind to make sure. Yes, there is a lot that we can do, a lot. But like I indicated, from the meeting that we had, the scientists, within the group, within the division, what came out clearly was with a fund, seed fund for generating, for purchasing consumables and chemicals, because chemicals and reagents are very, very expensive. This work, this proposal, that the request that this company have been brought to us, just a few chemicals, and I was showing the director the bill. It was around 17,000 Ghana cedis, and we are wondering, I mean, how they can, I mean, they will be able to, you know, pay, I mean, for us to just secure the chemicals, just the chemicals, nothing else has come, I mean, has been factored into it. So yes, it is our concern, and we as a team in the division, we are going to push ahead to make sure next year when we come here, there will be something tangible to. Yes.
I want you to touch on, you see, SIP everything, and the cost to us some machinery and other things. I want you to tell us staff here what is SIP left with us and what we can do with those machines and other things.
One for the breeders. Normally, I mean, for whatever traits, I mean, most of the traits that you look out for in the course of performing activities, there are some compounds, metabolites that we need to track. We have at the SIP lab near-infrared spectrophotometer that can be used for sugars, I mean, to quantify sugars, estimate the amount of sugars, check the oil, the fatty acid profile of the oil, and we have the RVA, which can also be used to determine rheological properties of starch for those that are working on root and tuber crops. There is a freeze dryer, a commercial freeze dryer that we can use. Basically, these are the cardinal equipment that they've equipped to us. So please, I mean, be aware that these equipment are there, and you can follow us when the need comes, I mean, for them to be used. Yes, your suggestion has been taken on board. We are going to pursue that. That is with the ISCA accreditation. We'll see how we can do. We can manage that. Dr. Seyi, yes, I mean, I am not aware of those equipment that you bought the seed division. I've not been able to visit this quite yet. I'll do so to ascertain their presence and discuss with OT how we can effectively use those equipment to generate funds. I think that is it. Thank you so much.
Thank you, Mr. Chairman. Prof. Adudapa, I used 3D quants for quantification of the protein, 3D quant method, and basically it uses copper to bind the group of your protein to generate color, and this can be read by spectrophotometer for you to do the quantification. Yeah. Prof. Betch, your comment is well noted, and I'll do better next time. Dr. Dankwa, yeah, I didn't apply fertilizer. I just looked at the common practice of yam cultivation, so I didn't apply fertilizer. However, I can also look into that. We can work on that too. And the point of my take on fertilizer application, I think once you deplete the soil of its nutrients, you need to reinforce it. So it's something that you can't. If you want better yields, then you need to reinforce your soil with nutrients. Thank you.
Thank you, Mr. Chairman. We wanted to know when the work will be completed. Yeah. The funds, because of the fear factor. Yeah, but what I can say is, if you take your food at home, probably you can minimize the hygienic condition. Probably they can boil water and then put it in the water, or they can treat where they do the pounding and everything. That one minimize maybe the microbial load. But as heavy metals, like I said yesterday, it's everything that we take. If they become foods, they are processed. The heavy metals is in the we are having it in our system. It doesn't reach maybe the threshold that we might be worried. So if the heavy metals, I can promise the director, probably see the yes, because what's happening is these heavy metals, you can, if you like, excrete of the system here. It bioaccumulates, it biomagnifies, and it bioconcentrates, and they have this affinity for the carbohydrate in the protein. So they say in your system, you don't they persist, just they persist in the environment. So director, if you take some, probably minimize certain things until I'm done with the work. and then profound how to take the samples from the residential areas. Prof, normally I would take the sample to where they do the mailing, in the mail, and I take the sample because they don't know what I'm after. Yes, so I'll just take it to those areas in the residential area. No, I'm not discussing. Yeah. Perfect. Okay. All right. So, from my, we also wanted to know which way to go, the pounding or the machine. But for now, I cannot make any pronouncement until my work is complete. But from literature I know, for now, me being an analytical and environmental chemist, I would advise that probably you pound your own food at home, or if you know a child that they do the pounding and they have hygienic conditions, then you can follow on it. Because the process with the machine, I am not for it.
Please, Mr. Chair, please can I speak to Dr. Autry is saying that briefly, please? May I speak, please? Thank you very much. I think Dr. Autry mentioned in his initial at the initial stages of his presentation that these machines are maybe hazardous except where stainless steel has been used to manufacture. So I think that you should incorporate into your study the proportion of machine fabricators who are complying, so it's not across that all the machines are using cast iron, or every machine is leaching heavy metals. Thank you very much.
Thank you. Yeah, and the risk, like I said, the risk is there. Even with even the thought of even running, not just even the need to, because they are all processed. So we need to check all that. Jonas, Dr. Jonas, I want to know about the element. Yes, I know the funds are inadequate, so I had to abandon the work. But I continued it in school, and then the publication is under review. So the work is under review, as the manuscript is under review. So the findings will come out pretty soon. Yeah, thank you. And Mr. Jumoku, you want to know? Okay, all right. Thank you.
Thank you very much. You asked whether we are involving food research in this week. no, there's no formal collaboration between food research and us. And with the school feeding program, we asked whether there is we've communicated with their director. We went there together with Professor Bo and Dr. Yeboah to see them, and I think last year they wanted to buy some for the academic year. Fortunately, all that we planted didn't go well, but we hope that we hope that next season, by God's grace, we can supply them some. Prof. Hans, please, you ask about the new methodology. Yeah, the new improved methodology, we have added cornstarch and then molasses, and then the brown sugar level, the sugar level has been reduced, and then the molasses the molasses fill that gap of the taste, so it helped the taste to be better, and it helped it to mimic exactly like the foreign one. But that one I have not done any analysis on it. That's why I did not present on that one, and that is what we have now for the new methodology. It comes out so. And Prof. Mariam, please, thank you very much for the comment. And about the water, we don't pour it out. We pour it away, we change it. We another one on it, we change it every 2 hours, we change it so that it will not ferment. We do that to prevent fermentation during the soaking. And even now, using the warm water to soak it, the time for the soaking, if you use the warm water, it helps it to become softer, and so the time for the soaking has been reduced. You can use about 6 hours instead of the 16 or 24 hours for your soaking of the beans. And then Prof. Lamte, you asked whether why don't you use the foreign one? My first check, I used foreign ones. The one with high tomato sauce, the color, the tomato concentration, be it reddish. That is the lily type, and then the hands. And that informed me to use the remedies to check which one will really do well. I used the lily and the hands, and it came out that the lily appearance and even the taste scored better than the hands. So I decided to prepare it. The hand doesn't have that sharp reddish color. It has that pale color. So I prepared I am using, when I was also preparing my samples, I decided to go back the two and see how best. But later on, when everything is successful, I will with the foreign one. But the foreign one has been to see which one people like, and I realized people like the lily type. So now I'm taking the tone of the lily type to produce our local one in the form of the lily type, and then after everything we will do assessment and check. And then the
Is nothing.
Dr. Rabero, the chewy and the hard, that one we communicated with the leaders. We are in communication with them every step. And then the meat substitute, Dr. Ankwa, you will see us for it. And then with the instrument for what during the analysis, our canteen was not involved. But I think since we are all here, we can introduce them to it. Okay. And the importation, Dr. Antivice, the importation of the cowpea, please is noted. But I think as said is seasonal. When we are able to meet that, it will reduce the importation of the foreign one. And I think our breeders are also working on the cooking time. And then Mr. Maku asks us to target the private schools. I think we started out with sweet potato and it went out.
No, that's not your question. It's for the institute.
Okay, thank you.
I had one from Prof. Hans on how to
Excuse me. Professor Sabo wants to make some comment on the school fee coordinator.
In fact, we contacted them to see whether they can use the beans, and after tasting some, they said it was good. But now we have to get our growers who are going to produce the grain for them. So this we are going to the north. We have seen we have contacted some agro who are ready to produce the listing before maybe we sign the agreement, because the product is even not on the market. And the second one, we have started also breeding fast cooking varieties. So we collected from CSIR with our breeding program, so that we are going to get fast cooking varieties in the nearest.
Commercialization, are you listening? Commercialization must be listening. Thank you. Oti.
Prof. Yeah, so my first on Prof. Hans question on how to improve our activities or the same time to ensure we don't lose out on our profits. What was actually happening last year was some people with some threshing machines. At the processing center, we do 3 main activities: threshing, drying, and cleaning. Now it was on the trend that some people bought some threshing machines and they were taking it to the farms of the seed producers and the other growers. And so it came to our notice. And from last year we had been working closely with a German company with our staff of director to develop a proposal to acquire some new machines that is more modern, that can help us in our activities, to help us improve the system. And then also, we've also done our best to put measures in place. For instance, last year it was very difficult even to get a tally card to monitor our activities, which pushed and directly assisted us to find a way to get some tally cards and other documents to help us in our documentations. So these are all the things that we are trying to put in place. On Prof. Mark's question on how to help breeders to our system, I think we will need to collaborate and work together. Sometimes it can be some materials to the cold room without putting it in a polyliner. They will just put it in a sack. So when we see such a thing, we call that breeder and inform that person that please, you have to put it in a polyliner, because don't think that once it's in the cold room, the conditions are regulated, so the can't absorb moisture, because these are hygroscopic in nature. So even in the cold room condition, don't put it in a polyliner, they absorb moisture, and when they absorb moisture, their metabolic activity increase, which tends to impact negatively on the seed viability. So I think we will need to work together so that we all see how best to help improve our system. Thank you.
They've done a good job, and this comment goes to scientific secretary. 5-6 people make presentation, and he said discussion should be only 20 minutes, 25 minutes. And the sort of questions that come, it will be difficult to even assign 5 minutes to a person. So I think next time we have to increase the discussion period a little. Thank you. Let's give them an applause, a hand of applause, and release them. My chairmanship position has come to an end. Thank you. Okay, PRO, over to you.
Thank you very much, Chairman Domoko. Our next channel is Dr.
Asante. Let's welcome Dr. Asante. Thank you very much, Pierre. The first person on the list is Prof. Queen. I've just been told you have 20 minutes because you also present the activities at the application.
Thank you very much. I'm Marianne Dorcas Queen. I'm with the biotechnology seed and food service as a biotechnologist, principal research scientist, a deputy director, and this is my presentation. Which one do I show it? Okay, so I'll be taking you through my 2020 activities, what I've planned for 2021, my publications, and my visits to CSIR CRI outstations. My first activity was with the USWA project, which is Gates Foundation funded through ITA, and it is collaborating with SARI, MOFA, PPRD, and private seed sector farmers. We have 3 companies, typically Hikma Farms, Fosua Food Chain, and Iribov. This is a project team, and the project outcome is to look at strengthening the pre-basic CDM system for the country using the improved yam varieties. And with that, we setting up temporary immersion bioreactor systems, and also having aeroponics and hydroponics systems to ensure high propagation of the pre-basic CDM. So for this, there's a lot of activities. So Dr. Amakwa and Mr. Abuaji will be presenting the aspects on tissue culture, production. I'll be talking of the technical backstopping we gave SARI and the farmers. Then Dr. Prempe will give us input on the indexing of the materials at the various stages, hydroponics and aeroponics activities are presented, and Mrs. Akom and Dr. Deborah will present on our field and nursery activities. So this is our hydroponic system that we developed for the project. So what happens here is, once we produce the materials in tissue culture, we plant them in these buckets, hydroponic buckets, and we get the bloom of canopy of fruit forming, and we harvest notes from here for production in the greenhouse and under nursery conditions. And I would say, because the multiplication rate is very high, we want to ensure that we don't pass any contaminated material to the farmers. So at the various stages, the molecular team come in, they tag materials that are suspicious, then they go on to the index molecular indexing at the various stages in the greenhouse, in the nursery, and on the field. We had a company buying some tissue culture material from us, and we from 2016 activities we produce quite a number of seed, so tubers, so we supplied to our stakeholders, including Professor Otu. These are the amounts we supplied. So we supplied over 1 ton of CDM. And we also produce the plantlets, the seedling for farmers. We produce more than 4,500 material for the farmers, because the farmers also have set up aeroponics, hydroponics systems. So this is what we produce for to take to farmers, the seedlings. So you can see a farmer who has come to pick up his material for his hydroponics and aeroponic systems. From our hydroponic systems, we have a single note for our activities. So these single notes, we nest them. These are the single note nests in trays in the greenhouse, seedlings, and also to produce some of them, the seedlings, when they grew, we planted them in boboya. The boboya that we see, so these are the boxes they come in, so we utilize them as planting to plant them in. We had to check the kind of compost material medium we used to plant them. We had a master's student on this, and we also have our own setup. The data will be presented later. And we from those systems, we harvested about 82 kilos of CDM, and this is some of the CDM that we harvested. So the seedlings we harvested, the single notes, we harvested over 20,000 single notes, established them in the nursery close to our field, and then we planted them out on the field. Some of them we in beds in the nursery to harvest from the beds in the field. So this is the field planting we were doing, and these are some of the we harvested from the single notes from the field. So from various sources we had materials we planted out, and we've harvested over 6 tons of CDM this season from November to January. So where to store the seeds was an issue. So looking around, we chanced on the stores. That the stores have a lot of space, so and it's all in a mess. So we found money, we cleared the place up, put some plywood to seal up any leaks, and we bought air conditions, placed them there. So the room is now very nice and cool, and we are keeping our seeds there. It's been very, very useful. And alongside, we've had PPRC come around to inspect our CDM before we send them out. The year under review, IT visited us to interact with us on the project, and these are monitoring visits to the farm. This farmer is in the north, and he is doing great with his hydroponic systems. This is the CSIR setup, and they in fact the year under review even helped them set up a tissue culture laboratory. So CSIR now has a functional tissue culture laboratory which they are using for the activity. This is a farmer in a company in Sugakope. In fact, when I got there, they had actually harvested most of their single notes and were already on the field. They had some materials on their aeroponics. This woman had her hydroponics full grown. Her aeroponics had already harvested and was planting them on the field. So for this, we are going to continue with all those activities to ensure that the system is running, the system can be picked up by the farmers and utilized very well. This is the last year of the project, and by the time we finish the project will be self-sustaining. We'll be having some field activities. Mr. Komo will tell you more on those. My second activity is with the Gene Bank platform project with the biotech team and Dr. Adolfo. This project is also ended this year, as we had some few activities. The main objective was to have the land resources of potato in sub-Saharan Africa collected, genotyped, and conserved and the long term, and also the viruses dry room was being studied. Virus detection used the small RSA, the small RNA sequencing. So we've worked with some 99 collections. 95 of them were what were presented by the Sweet Potato Program yesterday, and then we had Sierra Leone with their materials through SIP, which was cited here, and we've done the data analysis already. We've received our data. So these pictures of all the plants are in the screen house and in the lab as well. So this is the data analysis. Some of the analysis that have come out. These are Sierra Leonean materials. These are Ghanaian materials. You can see some interface and some materials that were used as checks in the analysis. We have a lot of data. We had an online training with DART last year, and we are going to sit down to write publication out of this study. So for the small RSA work, it's a STEM project. So it's in, and he's actually cutting out small RNA, sequence it to get the diversity of viruses that we have in the system. So this is the workflow that he's using. So now he's doing the sequencing, and we can detect the various viruses and catalog them. RNA, a good quality RNA has extracted, and with the analysis run we have the small RNA that are going to be used for sequencing. So this is the project is finished, but we are maintaining the cultures for prosperity. And then there's the INI project, which is also looking at yam index at the various cities. Like Dr. Yabua had a very not interesting email last night. Maybe they wait for you about something, they drop the bomb that they are finding is going to be cut. So we are not too sure. Dr. Pempo presents some aspects of it, but this is going to equip us with a molecular tool diagnosis tool, which is quick for sequencing your viruses. This is the workflow that we are developing. You say, why are we doing it? Because we are working with our young materials that we are tracking the status at every stage of production to make sure we are not passing out infected planting material to our end users. So one technique we are going to use is the main ion system, which is a powerful real-time DNA RNA sequencing device that's in your hand. You are in control of your sequencing data. This is it, and it comes also with a bundle as the adapter, which is very handy in analyzing your molecular data. So other activities are being teaching at the CST, supervising students and mentoring. These are my publications I dedicated last year to its cryopreservation book. I was the editor, which the book was looking at current advances and evaluation protocols in preservation, and then these are the publications I was involved in. So for the outstations, I'm very passionate about it as deputy director, and I think it goes to all of us that we should really take our outstations seriously. As an institute, director was telling us a couple of days ago that we are not even sure of the space that we have here. We might lose to one point in the land space that we have. We might be left with only our offices. Fingers crossed. But the thing is, if you go outside, you go to organizations, they just take you to their office, and when you have to go to the field, you have to travel miles to the field. If we are not careful, that's where we are going to be in the future. So if we don't take good care of our outstations, we won't have any place to put our research activities. So I was at Peve. They have 11 acres and at Peve and 20 acres at Gulukwati, and this is and even that the Peve land is supposed to be taken over by Mufa. So okay, I'll do that. And the team that we have there, and so how we have 904 acres. So how has it done? And I think they have only about 6 members of staff. Some are aged. We can see the water body over here, and that one too we have to be careful because Mufa, these are state farms properties that were they bequeathed to us. So somebody can also contend for the ownership, but we have the ownership now. And if you are doing any research in the Volta region, please send something there so that the place will come alive. In fact, this is their block of offices. They have a storeroom. They have some office space as well, and quarters are not being occupied by staff. Ministry of Education has taken over the quarters. In Pokwasi, we have 11 acres, which we are not sure of whether we want, because there's Mofa there, there's everything is happening around Pokwasi. This is the officer in charge and his cassava setup. Will be squeezed out of Idra, but the teacher Chrome, which has over 200 acres, the stream nearby, this is the staff. Idra is quite in course, and I say that you can do better at Idra, so please let's consider Idra. Akumadan has 25 acres, and also they are battling with Mufa. So for the space and everything, if we are not careful with time, we will lose it. I think for 15 acres, there's a nearby stream. It's I see it as a good isolation place if you want to do some beader seed production. They have some coconuts and oil palm trees which may need to be felled so that because it wasn't properly established. In Nasi, it's another hot spot. The MP there has to be sure of the seat. So we have over 800 acres of land. The soil research is also there. In fact, most of these places, when you go, you see either animal research or soil research also there. So we have to partner with our sister institutes to make sure that all these properties are well conserved or maintained. So I propose to direct that we can have a system ensuring that all these outstations are managed by researchers. If we can't have research at a place, maybe we can put one researcher responsible for Ejura Akumadan, another for Ohau PV, Pokwasi Asin for two. As we it shouldn't be easy connect two places with a if you have a project there, you can install on your project. Look, I want, this is where I'm putting my work. Water is an issue. So can we put a borehole, solar powered borehole and irrigation system? It won't cost too much. If you calculate your fuel alone, your TNT alone, you can get a percent that you use for that. Most of them have, even the Chrome has a stream that if we tap it, nobody should have issues when the rains fail us. And so I think I've talked mostly on all these. If you are organizing a field day and it's close to where our outstation is, can you do it on station to add visibility to the station? And I think we need to look into MOU with MOFA so that they don't, organization don't take over what we have. And big projects, we are looking at the resilience science project that Prof. Otu is leading, and we've developed something to take to the outstations. So your activities to take something to the outstations so that we can fully equip the outstations. They need computers. The director is has put in place some things to ensure that the computers and NAPCO and service since I think some of them have to go to our outstation. So thank you very much for your time and your attention.
Give her a big hand.
Good morning. I'm with friend here with the molecular biology unit of the BSc division. I'm here this morning to present to you activities conducted last year and proposed activities for this year. Activity 1, training on data management and analysis using the RSS Scout software. Under the Next Gen Committee of and Partnership Projects, we had a 5-day training on data management for scientists, technical staff, and students of the CCC. 27 participants made up of 15 males and 9 females benefited from this training. These were some of the pictures that were taken during the training. Activity 2, genotyping of SNP germplasm using genotyping of cassava germplasm using SNP markers. The Cassava Improvement Program over the years had a repository of cassava germplasm, but along the way we lost all those materials. So under the project, we decided to collect some cassava germplasm from PGRRI and from some farmers. These materials were genotyped with 2 sets of molecular markers. The first was a quality control marker which comprised of 18 highly informative SNPs which are used for routine fingerprinting to determine genetic similarities and also identify duplicates. The second set was a trait link marker for traits such as the gene resistance gene, grain mite resistance, provitamin A content and dry matter content. This table is showing some of the SNP markers that were used and then the traits that they targeted. We have received the genotyping data which we are yet to analyze, but then this is just a summary results of the genotyping data that came along with the genotyping data. For a SNP, when you use to do your genotyping, your sample will have either of 2 alleles. So when you look at this table, like the SNP number 001 with a plate number 5 consisting of 96 samples, what this is telling us for the first row is that for the 96 samples, about 97% of the sample had the allele C, and about 3% of the sample had the allele A. So that is just the summary results. We are yet to analyze the data. Activity 3, under the NRI CDM indexing project titled Using Different Diagnostic Molecular Tools for CDM indexing at various production stages. Yield production has compromised by a high impact of viruses as well as the unavailability of new planting materials. And this is a serious problem that we need to work on. Currently, the proponic system that we have has a high multiplication rate and has the ability of producing thousands of plantlets from a single plant. But the problem here is that the virus titer in these generated tissues are generally low, and it makes virus detection very difficult. Currently, there are strategies that are more highly sensitive than what we are currently using, and one of them is a high food sequencing using immune. The aim of this project is to improve the capacity of yam virus diagnostics by incorporating the high throughput sequencing method at different points of the seed yam production process. This project has a lot of capacity building activities, but unfortunately, because of the COVID, we were not able to do much. What we were able to do last year was to send some samples to our partners who are using those samples to establish the protocols. Once the protocols are established, they will share the technology to us and we'll start using it in our lab for virus indexing in yam. Activity 4. Demonstration of tissue culture generated cassava planting materials. Last year we were fortunate enough to get some funding from MAG, and 50% of the funds that we had to go into supporting our ISO accreditation activities. The other 50% we used to conduct this activity. The vegetative nature of cassava coupled with the continuous use of the same planting material year after year leads to accumulation of pathogens, especially viruses, and this leads to reduced yield reduction on farmers' field. So to enhance the productivity of farmers, cassava varieties have to be cleaned frequently to produce high quality planting materials. The objective of this activity is to evaluate the performance of tissue culture generated planting materials and also to generate new stocks of high quality clean cassava planting materials that will be subsequently made available to farmers. So what we did last year was establish 2 cassava fields, one at Fumeso and one in Mampong on a farmer's field. And from these 2 fields we have about 15 cassava varieties, which is approximately 1 acre. Data is being taken and we will analyze the data to determine their performance. The image here, this picture was taken when the planting materials were being prepared in the tissue culture for field planting. This is at the time of planting, and now this is how the plants are looking on the farmer's field. These are tissue culture generated planting materials that we used. I was also involved with a number of helping and assisting students. So we have students working on gene expression, assisted selection, molecular characterization and diagnosis. We also collaborated with the CSIR and the pharmacy department of KNUST, where they use their facility for their work. When they come here, we charge them for the bench that they use. They bring all their reagents and consumables. Let's use our facility. So we charge them for using the facility. This is just an example of a student's work that the student worked on gene expression, and she was looking at differential expression of genes in selected yams that were subjected to aeroponic system. In time, the student is going to present a seminar on this. I was also involved in these other activities. My proposed activity for 2021, the first activity will be training on using our software for analyzing genotyping data. And from the genotyping that we did, we have generated a lot of information. And to make good use of this information, we have to be able to analyze the data. So we'll be having online training with IITA, where technical staff will be trained on how to analyze the genotyping data. Once the genotyping data is analyzed, we'll be able to identify and remove duplicates from the core collection, from the collections that we took from PGRI. And also, because we use trait-linked markers, we will also be able to identify that some germplasm that carry specific traits such as DMD2 resistance and dry matter. These materials, because we use genotyping, we are supposed to validate whether what we are getting from the genotyping data is right or wrong. Activity 2 will be screening of cassava germplasm for end-user preferred traits. And from the preliminary screening that we did from the materials we collected from PGRI, they were very diverse, so there is a possibility of us having some important end-user traits from it. We want to screen this material so as to be able to identify germplasm with end-user preferred traits that we can incorporate in our cassava breeding program. So we're going to select 30 cassava germplasm, which will be established at FIMESWA using a 6x3 alpha lattice design with 3 replicates. These are some of the data that will be collected, and it will be analyzed using our CSIR software. It is expected that at the end of the screening, we'll be able to identify the cassava germplasm with end-user preferences that will be used for further studies. I'll also be involved in these activities this year. These are my publications. We are grateful to our sponsors, collaborators, and our supporting staff, especially the National Service personnel and the NAFCO trainees, who, when during the lockdown, we camped them here and they did a wonderful job for us. Thank you.
Considering a lot of color of your sports, it needs to watch by. Some chance, you can pick over the problem now. Thank you very much, Prof. Dr. Chairman. My name is Charles from Biotech and Post Service. I'm a Commanding and Biosafety Officer. The outline of my presentation will take us through the respective down to acknowledgement. This is the list of my activity. By way of introduction, yam is a killer crop with economic benefit, or in helping in poverty alleviation and food safety. Worldwide production of yam is estimated at 73 million pounds, with majority being produced in West Africa and East Africa, Ghana and Nigeria, where majority use safe seeds acquire as inheritance buy from other farmers. Activity 1 is a seed multiplication trial using adaptive yam techniques such as the aeroponic generated seed, and the objective is to establish functional, scalable, sustainable system derived by private sector investment that delivers sufficient quality of high quality seed of quality or improved variety to farmers at the right time and at the right price that encourages adoption. These materials were used as seen from the pictures, where mini set, the single note, the multiple boxes that Prof. about, and this happened to be hydroponic plant establishment in the greenhouse. Beds were used using the adaptive yam mini set techniques, where 30 grams of the mini set and some root cuttings that were placed in our measuring with a planting distance by 100. Our target was to produce 40,000 tubers. At the end of the trial, over 40,000 were produced. Year after year, it was realized that the surviving rate of these
yields in our nursery were not up to 100%. This recorded a mortality rate of 30 to 40%. Then an investigation to compare the systems, vines from the two systems on the surviving rate, growth and yield of the different single nodal cuttings from aeroponics and hydroponics. And my objective is to determine the surviving rate of the different sections of two varieties from the hydroponics and aeroponics at the nursery stage, and look or investigate into their growth and yield of these sections using these same two varieties from hydroponics and aeroponics system. TDR 95 by 1977 and TDR 89 by 0265 in the experiment, where people were having the length taken and cut into 3 different sections. These sections were planted separately from each other in the nursery using an experimental and transported into onto the field using RPD 2x3x3 in 3 replications. At the end of the experiment, it was shown that vines from the aeroponic system with TDR 9519177 perform better as compared to vines from the aeroponic system. They still perform better than the mid section and the end section after 6 weeks after planting. Figure 2 represents average number of leaves from week 2, week 4, week 6, and it's shown that the aeroponic system perform better than the hydroponics. Figure 3 represents the number of seeds, and a similar trend was recorded among all the parameters that were taken in the experiment. Our collaborators recommended 250 gram weight of yam for our yam production, and an experiment was set to look at how we can to confirm or debunk the weight that was recommended. The objective is to confirm the recommended weight for where yam production using different weights. So 250 and 335 were used in the experiment with TR 1919177 Kukupa and Manchis in an RBD experiment, and it's shown from plate 14 with full establishment how they perform and at harvest. And the results, it shown that Manchis performed at 375, but other varieties shown less significant among the 50 and 375, and it was shown from all other parameters like the width of the tubers. Commodity which I worked on have this receptive, that's the growth and nutrients, and here I was looking at iron and boron uptake response in granules. Granite is an annual legumes and an important cash crop in Ghana. And why iron? Iron involves in the synthesis of chlorophyll, and it is essential for the maintenance of chloroplast structure and function. And why boron? Boron is required by plant and it is higher for reproductive growth, so it helps in pollination, fruit and seeding or seed development. And the objective is to develop the effect of combination between the foliar iron and boron on granite growth, yield and nutrient uptake. The materials used were DA and Yenyaso in an RCBD experiment with 2x3x3 pictorial planted distance with treatments, and were established in Fumasa and Komadan. Data were collected and analyzed using the STAR version. And this picture shows how the fields were established, how application was done, leave area data, and the exposure of models as a result of draft where we use water holes in irrigation in our fields and field establishment before harvest and leaf area take. The results shows that the granules responded positively with iron at 90, with boron, and the same was recorded at this represent values from Funasa and this Commodine, and likewise for the total pot yield where Yenyaso responded positively at iron at 90 and boron at 22.4. Funasa had 820 and was had this treatment, that's iron at 60, boron at 22, and the trend goes showing that the treatment that was applied on the granule had positive effect when it comes to especially chlorophyll content, which iron really enhances. But safety activities, I was involved in safety activities with the CSIR, and we look at management when samples were received, and here is the PI in his one pack, and field establishment, management, visitors visiting our fields, and the management at the gate, and he's in rating here. You can see Prof. Again, we held the KNUST medical school to acquire a permit for a GM trial. We also helped in the disposal of hazardous from COVID-19. I was also involved in COVID-19, where through my lovely we were able to get nose masks from RST and the provision of Veronica Bucket and installation of automatic dispensers. We were able to supply 3 companies with materials, and field establishment was done in their fields and open day. I had this as my challenges, and you can see the deputy director coming together with the driver to help us in our field experiments, and we had to sometimes use water to water our fields in order to lose our experiment, and here water tanks which got most of our nodes exposed. This plan activities want to repeat experiment that was list of applications. Extension leaflets were also prepared for care, and I want to say thank you to Yusua and the team, legumes, new management of CRI. Thank you.
I now call Ms. Agnes Echambo to the representation.
Good morning. I'm Agnes Echabwaji, and this will be my presentation outline. I will present the activities I undertook in the year 2020, and then my planned activities for this year, and my few list of publications. This is my research activities undertaken in 2020. My first activity was cleaning of young plantlets using meristem culture and chemotherapy. Plantlets were kept at a temperature of 20 it for 4 weeks and then transfer it to the therapy machine at a temperature of 36 degrees for 16 hours and then 32 degrees for 8 hours. The light intensity was 500 watts. The relative humidity was 70 degrees. And then afterwards, in vitro mechanisms were cut. Far, these are the varieties that we've been able to clean. They include CSIR, Kukupa, CSIR, and the rest. My second activity was an evaluation of some selected varieties which I undertook in my objective was to detect the growth of different tissue culture generated planting varieties for upscaling. My planting materials were matches CRI Kukupa and then DR 19177. For my culture medium growth condition, I used single notes and MS media supplemented with this concentration of carnitine, L-cysteine, A-disulfate, sucrose, and then agar. So what I did was that I dispensed 50 ml of the medium into test tubes and then autoclave them at a temperature of 121 degrees for 15 minutes. It was a CSIR the entire replicate experiments. The data I collected the number of leaves, number of nodes, number of branches, and plant height. So for the growth rate, as you can see, there was significant difference in terms of the number of fruits, but there were not significant difference as for the number of branches. So my conclusions are that growth rates of young varieties will be established, and then other growth regulators will screen to optimize the performance of. So my planned activities for this year, my activity will be to do a comparative service study of temporary immersion bioreactor systems, conventional plant culture that we already know. As by way of introduction, yam is a starch food and forms calorie intake in main household in Ghana and sub-Saharan Africa. Ghana is of course the leading exporter of the crop. In production of planting materials, using the liquid medium has been considered to be more effective than the conventional because of the fact that access to the nutrients, and then also the ease of handling and the comfort of upscaling, and then also automation. So the plant materials will be TDR 1917, CRI Kukupa, and my culture medium will be I'll use the same MS, and then I'll supplement them with these concentrations of sucrose, agar, and then I will dispense 10 ml into 30 tubes. I'll add agar to make the medium solid, but for the liquid medium I'm going to use the two bioreactor system that you already have running in the lab, which is the Rita system and then the plant form. For the Rita system I will dispense 30 ml of the liquid medium, 30 plantlets. For the plant form tips, I will dispense 300 ml of the medium into the vessel and it will contain plantlets. I'll autoclave them, pH and autoclave the medium. So my plant materials will be 191 CRI Kukupa. Another aspect of the work is so another aspect to compare the growth rate of the different varieties using different immersion systems. This time around I'm going to use one temporary immersion bioreactor system, which I'm going to and I'm going to use a 4 flash times, 4 different immersion times. My first 1 minute flash time, 30 minutes rest time, 1 minute flash time, 60 minutes rest time, 1 minute flash time, 90
flat time and 120 minutes rest time, it will be a CRD experiment 3 by 4 factorial 4 replicates. I'll wait for 15 minutes. So I'm expected that at the end of the experiment, rates and stable emerging times of each variety will be established to automate the growth of plantlets for mass production. My second activity will be screening and identification in vitro yam variety for micropropagation. Yam is affected by a lot of pathogens and apart from the fact that the bacteria causes a lot of yield loss, they are because they restrict international exchange of germplasm. Plantlets, 40 samples of yam, and then the root stem and the leaves, 8 old plantlets will be chopped on incubate them at a temperature of 27 degrees for 7 days in darkness. Afterwards, incidents of endophytic bacteria will be identified and recorded. My third activity, I'll still continue with the cleaning and multiplication of yam varieties using plant tissue culture, temporary mesh reactor systems, and then the VPAC. These are the thank you.
Those of us who are stemless, you have to give us double portion of the trapping. Now call the policies.
Thank you very much. Good morning, everyone. The first activity I was involved in is the development of Ghana's tomato industry. And the project implementation area is good. Here is our research team, and by way of objectives of stakeholders in the tomato sector on improved production and handling practices with the value of fresh tomatoes, because here in Ghana, fresh tomato carries a premium value over processed tomatoes. Consume a lot of various forms of tomato. The other objective is also the overall overriding of along the supply chain. In terms of post-harvest processing value, in spite of the fact that we import the world's second largest importer of tomato in various forms, second to Germany, we do locally, and in terms it's a yeah, that's the losses in terms of production costs. So why such losses is a big question. We spent phase 1 of this project doing so many, and we came to a conclusion that a collection of various strategies which we can. Some of the challenges are linked with limited packaging and handling logistics, poor infrastructure, which we can do nothing about. But we also realize that a large proportion of us in phase 2 well known that the quality of fresh produce cannot be improved after harvest, and so tomato quality at the time of harvest has a great bearing on post-harvest losses along them. Traditionally, Ghana's tomato production is characterized by direct contact of with the background of pests and diseases. So we devise very simple and basic to address these challenges, seeking a mulching. When we assess the disease incidence of action methods at demonstration sites, we realize that of tomato yellow leaf curl virus incidence and the other lower. But when we look at the cilium wilt, I hope interventions we are introducing seem to fare this particular one, where we go without the traditional wedges and we mulch. So in 2020, at the latter part, which involved farmers and extension agents from our project area, also another area that's Kumadan, and the aim was to expose them to these production post-harvest quality of tomatoes. In terms of yield, we were surprised to find that rate of 7 harvest each plot. This is kilograms per plot. This instance also had a fairly higher yield than the other interventions. Here is farmers and extension agents doing their own demonstration site. The director was not in this location. We also observe differences in appearance and market value, quality in general. The different using the different production methods. In this over 3 seasons from 2020, we concluded in 2019 and one in 2020. From a previous study, which was 4 storage, 7 days, measuring weight loss after quality intervention, less was weight loss, telling us if placed within the marketing cycle. So we are concluding the best seasons of studies. The best version is when we mulch on flat ground without reducing mistake. This is good market value and short life. Thank you. So this is in the Papa release facilities beans varieties. The ultimate goal of this is to stimulate demand for the release of commercial production in a sustainable manner. Initially, in place of company in well-known indigenous Ghanaian dishes, we have diversified product concepts through the development of products. And so we came up with the mixed baked beans and vegetarian milk. We are trying to disseminate the information. We engaged in a one-day training. They were drawn from CSIR, National informal youth employment, SDA hospital canteen, because of the vegetarian interest, was still kitchen matrons. So some of these pictures have already been seen. Some of the meat substitutes, and they are very cheap compared to the real meat. 100% product in here. This is a little bit of animal product with 8%. This is half. 100% from a former variety. It's very tasty. Okay, so this for the vegetarian milk. For this, so we intend to train the youth in value addition of tomato products. That is the drying of tomatoes hygienically. So for the youth training, I the area that's the aspect is really going, okay, why drying of tomatoes apart from contributing to, it really opens up niche markets, because elsewhere it's the products ready to cook. In various stores and restaurants, I don't see some in Ghana, but elsewhere they other specialty foods. And it's also coming up as ingredients in the cosmetics industry. Put on this program. And the problem being, we are in the process of looking that which we will be careful to launch with the library when it's completed. Malaysia is not here. We organize at least one exhibition to showcase the local and innovative products from this with media engagement. I have three journal papers to acknowledge Abdullah Isaac, a national service personnel who have been instrumental in our progress even throughout this period of trials. Other NSP service personnel and macro trainees to acknowledge, and I am indebted to the director and the entire management. Thank you very much.
For this session,
my name is Victor Amakwa, and I'm in the biotechnology and post-harvest division. I'm a tissue culture specialist and also a plant. Yeah, so I'm going to give a presentation on I was involved in 2020 and planned activities for 2021. A list of my publications and acknowledgements. For the activity 1, we are going to mass propagation. You know, Agnes presented on materials being cleaned, and mass propagate them, and you have to acclimatize them so that the hydroponics and the aeroponics systems. Yeah, so some introduction. You know, like we're doing the conventional planting, and with the USWA project, a new system was introduced. Immersion bioreactor systems. 2 brands. We have a platform and like Agnes said, this one is
much more efficient than the conventional. The nutrients are directly in contact with the plant, and it helps prove. Yeah, so we had as our aim to disease of yam plantlets to feed aeroponics and hydroponic systems. So after the materials were cleaned and they were certified, prepared meat, them using our yam standardized protocols, and the materials were incubated for 3. After the materials had grown for some time, we also, against subculture, we had to apply. We use 2 systems. We have what we call the VPAC system, and we also have the seed trays acclimatized so that they could move out of the tissue culture laboratory. Some of the results. This table shows a list of and the quantities that were produced in different systems. A total of 41,278 plantlets, 360 from tips, and 518 additional plant tissue culture. Yeah, so after the material using tips and CPTC, we had to, so like in this, 5 varieties were used, TDR 9519, applied 7,268 on 15,207 on using seed trays, making a total 2,475. Yeah, as a way of summary, in CRI TC as usual wants it to be, and that helps us to meet a target. The VPAC system and seed trays were efficient for acclimatizing. Plantlets obtained from tips and subsequent hardening using VPAC and seed trays were able to produce enough hydroponic systems. Yeah, I was also involved in the CSIR fund, under the CSIR 2 revolving fund sheet. Lab and also at the screen house for like we did some plantain multiplication and hardening, and this is the research team. Yeah, so during the previous years we realized there were potato planting materials, but most of the time, okay, thank you, like people wanted, it was of high demand. So we learned from the yam experience. We mentioned by reactor system. So that helped us to increase our in vitro base them in the screen house. So in the year under review, we were able to produce 37 in vitro plantlets, and we were able to sell 220 pre-basic seed vine cuttings, and we're able to generate 37.88 Ghana cedis. Yeah, so we did similar thing. Planting, we do only the in vitro multiplications. We're able to generate like 1,004 plants, like 1,450, and we're able to sell about 1,050. Yes, for 2021, we are going to of clean planting materials, and our target now is 62,000 pre-basic seed yam plantlets. Put a student on a project, and we are going to do evaluation of temporary immersion bioreactor system and assessment of. Yeah, so we are collaborating with KNUST, and this is our research team. Yeah, so like, you know, yam is a vegetatively propagated crops. We want them to advance, and so we grow them in vitro. But then when we grow them in vitro, they also produce their growth, and want to investigate this for so that the yam can grow very, very. So our goal was to improve the performance and rapid multiplication of Dioscorea rotundata using tips to examine the growth of local and improved yam varieties, and also to assess phenolics exuded by yam plantlets. Yeah, so the location will be at the TC lab and biochemical lab. We're going to prepare media with varying levels of growth hormones and activated charcoal. Our plant varieties, because we know they exude more phenolics and one improved variety, and we will standard conditions. We also perform high performance liquid chromatography to identify the so we are going to use 3/4 factorial and we'll set SD at 5%, and these are some of the data we are going to take: survival rate, number of nodes, root development, phenolic content. Yeah, so it is expected that best concentrations of IBN for the rapid growth of local varieties, and also we will determine the effect of phenolic exodus on yam. Yeah, for 2021 session, the demand keeps on coming, and thanks to the breeder, Dr. Adolfo, for very. So like we are trying to expand so that we can meet the demands that will be coming. So Isaac left us house behind legumes. We are prepared, which will grow some more sweet potatoes, and our target is 200,000, and like we improve plantain production both at the greenhouse and the levels. So we are to be submitted to the CSIR grants. So I'll be involved in these are a list of my publications, and I would like to be working technical staff, NAVCO and National Service Specialist.
Let's take a seat. And then we all know that Ekasa na asono then we come to the wise men and direct. So I want to see the